Okay, here we go. Thank you for your patience with the arduous task of switching from the Mac to the PC. So this was when I was notified of the award, I spent a lot of time thinking, what should I talk about? I looked at some presentations by other awardees, thought about what I thought was interesting, what you guys might think was interesting, and I ended up thinking, I've been involved in lots of studies, a whole large range of topics over my career, and often it leads to the point we think, this is really a cool idea. There ought to be subsequent studies. It is a tool ready for real time. And those of you who are faculty members, you have students that you can pass on ideas with and share things with students. As a university, at USGS, I don't have that same opportunity for students. So I thought what might be interesting is to share with you some of a range of isotope studies that I've been involved with over my career, where I think there's some cool next steps that could be easily undertaken, and I'm sharing these ideas with you. I'm calling them low-hanging fruit, things that are just obvious places to run with the science. And I hope that turns out to be interesting to a large range of people. So I'm going to start with my very first study because it's still warm in my heart. This is Vance Kennedy contacted me and said, oh, you know, we have this watershed in the atoll of California. We're trying to figure out where the water's coming from, and this is something isotopes are used for, blah, 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 blah. Would you be interested in collaborating with me on this? And this was back in the early days when I was not very aware of isotope hydrology, and I thought, geez, Vance, it's raining, the water is rising. People are really wondering where the water is coming from. It's obvious it's coming from the rain. And it turned out it's something so fundamental that everybody's little friends down the block would say, of course, this is a known. It's not always the case. It was one of these, it's immediately obvious that it must be working this way, but it doesn't always work that way. And this was something it was good to learn early on, is that a whole lot of things that you might think are obvious or you think you know what's going on might not be true at all. You should be open to the possibility that a lot of things you think you know aren't. You're wrong. So I often use the phrase, half of everything you think you know might be wrong, and the same with everybody else. It's a useful starting place. So I put this one up just to remind me where I came from and how valuable it was to learn that lesson early on. My favorite water isotope study is a large-scale study that I undertook with Tyler Colman, and I thought I would run through that quickly because I think there are some interesting ramifications of this study that I thought people might be running with. I thought we would be running with, but it hasn't happened, so I thought maybe these things might interest some of you. So back in the 1980s, we were wishing we had a better set of data of atmospheric water, O18 and delta D, in rain samples. The USGS did not have a rain network. We had surface water networks. So Tyler and I thought, well, maybe based on the work of Irving Friedman and others, streams, particularly small ones, probably a pretty good proxy for long-term precip records. So let's try to piggyback on the NASQUAN program to develop isotope maps of the United States. And back in those days, there were 600 sites being sampled about once a month by the USGS. We picked 400 based on all sorts of criteria, and we sent bottles, people sent us the samples, and the rest is history. And it's still one of my favorite studies because it works so well. If people, now you go, well, it's obvious, you make, this, of course it would look just like precip, but we didn't know that at the time. There had been nothing like it ever done, and those first contour diagrams when you went, wow, this really shows the kind of patterns from much smaller sets of data people are seeing in precip. This worked. So I show the delta 18 values, but we have the delta D also, and it was a very important study that we thought would be a first step toward using isotopes to better understand how all of these rivers worked. And it didn't actually turn out that way, and so that's going to lead to some low-hanging fruit. This river study of ours and some smaller scale but longer term studies in Europe were the early prototypes of the grand vision of the IAEA's GNIR program, Global Network of Isotopes in Rivers program. When I was meeting with them a couple years ago, it occurred to me that although these data have been published and available for 10 years, they haven't really been used for the kind of water quality, watershed hydrology studies that we originally anticipated. Instead, their main uses have been to provide empirical support for algorithms that interpolate precipitation data over large scales, most notably by Gabe Bowen, and a whole variety of uses for this for human, animal, and climate change forensics, but not the kind of applications we thought about. So I would like to encourage more hydrologic investigations of these data sets, so I put some ideas of things that might be useful to try. And I just threw this one up here. People who are going, oh gosh, there are all these sites where you could get modern samples. Sadly, that huge set of monitoring sites has been greatly reduced, so that the USGS NASQUAN program that used to have 600 of those sites, some of them have been passed on to the NAQUA program, and some of them they've been passed on to the states to analyze, but we don't have that huge set of monitoring network sites anymore. But there are a whole bunch of other studies across the United States where people have piggybacked on a variety of programs to get water isotope data. I put this because there's a huge amount of data out there where people have done isotope studies and interpret the isotopes, but haven't put the data together with all the chemistry and hydrology data that are available at such data-rich sites like the USGS, NASQUAN, and NAQUA sites. So back in June, when Jeff McDonald gave his Kawasi talk, that the sentences at the top, he put in one of his slides from a paper by Rich Alexander of the USGS. And I wrote a note to myself to talk to Jeff about this, but I never got around to it. But one of my puzzles that I had for years is, despite this statement that the headwater catchments are where most of the water and the nutrients are coming from as they work their way into the big rivers, the isotope data don't really show that. The isotope data look like the water there is from local rain there. And it seems to me that there is a disconnect here that somebody ought to spend some time looking at. So I put this up, Jeff, this is something we've talked about, but so low-hanging fruit, these were just some ideas of ways forward to compare the isotopic compositions, catchment size, location of sampling site, yada, 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 with the isotopes to try to figure out why some sites are seeing just what would be the local rain, and other ones are seeing rain from further upstream where the water is coming in mostly from the first and second order catchments. Why? Why some, not the other? Does the river water composition mainly reflect the headwater catchment or not? What are the discrepancies, et cetera, et cetera? You can read these as well. But these are just some ideas of things that are puzzles in that huge set of data and are probably there in the larger sets of data that other people have gathered. There's a huge amount of data here of hydrology, isotopes, and chemistry about how these river basins are working across the United States. And it just seems like there's a real opportunity for someone with good modeling skills and who likes large data sets to try to make a step forward from empirical data. More low-hanging fruit, and I'm going to zip through it because time is zipping on by. There are lots of puzzles about how the water isotope data showed a different evaporation signature than I might have thought. Was this evaporation in the lakes? Was this evaporation during recharge? What does this mean? Why are some rivers more evaporated than you would have thought based on the residence times? Lots of puzzles here. At one time, the thought was that the IAEA's GNIR program would continue with water isotopes as a focus of something to encourage countries across the world to develop monitoring sites. But in the last couple of years, they have changed focus and realized that the big problems in these countries is often too much nutrients, how these nutrients affect algal blooms in rivers, and a big monitoring program should be focusing on nutrients and organic matter and sediment, things that have more direct, immediate relationship to human problems. So if folks are interested in large river systems, there's a whole group of people across the world who have wrote proposals to work with the IAEA on this, and most of them are happy to have more partners join them in trying to better understand our large global rivers. That study was my first foray into piggybacking on federal and state and local monitoring programs. I started my career early with Jeff McDonald and Vance and the Webb watershed people and a bunch of people doing lots of fun watershed hydrology and biogeochemistry studies. But in the early 1980s, the early 1990s, where there were significant cuts to federal science, it caused a lot of us to think about what were the kinds of science that are best done by the USGS, and we could do them better than university scientists, and this is a niche we ought to be occupying. And this caused me to kind of move my focus to larger scale watersheds where there were larger numbers of humans and major environmental impacts that could be studied with isotopes. So it's been a fun transition. So there are a whole lot of reasons why you would like to piggyback on big programs. One of the obvious ones is that somebody else might have collected the samples, they might have analyzed some samples for chemistry, lots of things that you don't have to do because you're collaborating with a large group. But one interesting aspect of it is this allows you to test hypotheses at such a large spatial and temporal scale and with such huge numbers of samples that a lot of environmental noise is reduced and the effects of large-scale processes are more apparent. This wasn't obvious when I started this foray, but as I look back on it, a number of the studies that we've undertaken, if you had did them at the small scale of futzing about processes and measuring samples, there are just too many places to collect a sample, there's so much variability, how do you get to the big step? So an alternative is to start at the big scale, collect samples, and make a kind of a leap of faith. This I sometimes call assume a spherical chicken kind of a model, where you assume that the variability blends together and the processes can be more apparent. It's worked so many times, I wouldn't call it article of faith, but certainly that I've not stumbled and fallen from. I'm going to talk about this a bit more, but my favorite example I'll talk about later is an atmospheric nitrate study that Beth Boyer, Doug Burns, and I dreamed up in the early 2000s, and I'll talk about that later. So this is my last strictly hydrologic example, and I mention this one because I think this was a tremendously important study with implications for serious environmental problems and a cool hydrologic tracer that we hadn't realized. So this was part of the PhD work of Evelyn Hinckley at Stanford, and she came over and said, Carol, we're looking at sulfate concentrations. What about isotopes? And so I got involved in this. Where do I start with all of this? The basic idea that makes this work from my perspective is that sulfur isotopes can be a useful tracer of agricultural sulfur additions. And elemental sulfur is a major agricultural chemical in California and most of the US. It's added largely as a fumigant and a pesticide. This is besides the huge amounts of sulfur that are added as soil amendments and for plant uptake. Plants need about as much sulfur as they need potassium. It's needed to build proteins. So it's a huge additive. What turned out to be cool is this source of sulfate had, in two studies that Eve conducted as part of her dissertation, a distinctive delta 34S value that isotopically labels bypass flow. And I remember in the early days with Jeff McDonald, where we were using water isotopes to trace new water and old water, and thinking, gosh, I wish these would be useful tracers of flow paths rather than where the water is coming from. Well, we have a tracer. This one, in these two studies, labels the bypass flow water, and it is distinctively different than the matrix flow water. So you can quantify these, something that you can't do with water isotopes or other tools we had. And the important part of this from an environmental perspective is this study showed that you could calculate how much of the water applied to a vineyards wasn't needed by the plants. The plants never got it because it bypassed the root zone and went on its way. And that's water that arguably could be saved by improved irrigation practices. So in a world, including California, that's facing more frequent drought, a simple method that would let you better use your water supplies is a smart idea. So I'm going to walk you through this idea. This is experimental design that Hinckley developed, where she had lysimeters that would collect the matrix flow through the soils, and other lysimeters that would collect the bypass flow. And it turned out, this is how this works, they spray native sulfur all over everywhere. That sulfur drips to the ground. There is a population of sulfur oxidizing bacteria everywhere. They happily make sulfate out of it, and that sulfate moves through the soil. And you read the paper, in these cases, it was nicely distinctive from bypass flow. We don't know if this is true everywhere. It was a study that I view as a pilot study deserving of future more studies. But this difference right here, but where the values for the bypass flow were 7.7 per mil compared to 14.5 per mil for the soil water, that's a huge difference, making it a useful isotope tracer. Here is a page from the WRR paper that shows how these calculations were made. So why do I care about this besides it's a cool tracer of a flow path that we didn't have before? And this is because I remember the acid rain years. I was a young scientist in Western Virginia back in the 80s and 90s as the Clean Air Act came into effect, and we were cutting back our sulfur additions. We know what the effect of that sulfur had on watersheds. Places where there was high concentrations of mercury in the subsurface and the right amounts and kinds of DOM, you got methyl mercury production. And the little diagram at the bottom shows a report by Mark Marvin de Pasquale showing that in the San Francisco Bay Delta area, places that have the right concentration of sulfate and have mercury there, you can get the mercury being methylated. The plot in the middle is a map of Napa County showing how much sulfur is being applied to these watersheds. It's a huge amount, and it's added to all kinds of plants all over the place. So the thought, and this is a quote from a subsequent paper that Evelyn and Pam Matson published, that this was not an unintended anthropogenic deposition of sulfur like what happened with the acid rain years. This has been purposeful, added in order, it's a less toxic material, is a fungicide and pesticide, but unanticipated consequences abound. This is the same kind of mechanism that resulted in the high concentrations of mercury in the Everglade. It was a substitution of adding native sulfur to change the soil pH so they could use a more slow-release fertilizer that ended up with a huge concentration of sulfate moving down through the system. So is this happening other places where, all across the country and other countries where a lot of sulfur is being sprayed? So I include some slides from some of the work the USGS did in the Everglades, making the point that there is a sweet spot, a Goldilocks spot. If the sulfate is too high or too low, you don't get methyl mercury, but at the right spot, you can get a significant methyl mercury production if there's methyl mercury. This process also works on a variety of other kinds of metals that might be in the soil or in wetlands. In places that have had lots of mining or burning of municipal waste, there's lots of opportunities to have high concentrations of toxic metal, which could be mobilized by this unanticipated consequences of adding extra sulfate to the environment. I think this needs further study with some more pretty plots. So low-hanging fruit. Two ideas. Is this do we often enough get a distinctive difference in sulfur isotopes between the matrix flow and the bypass flow for this to be a useful tracer of a flow path and for a potential savings of water with improvements in best management practice? And the second one is this excess sulfate being supplied by using sulfur as a fumigant? Is it in places mobilizing metals in adjacent wetlands and riparian zones, setting ourselves up for environmental problems to come? I think this is maybe the coolest thing in my radar. If I had 5 students and somebody was interested in this, I would go for this one, because this is really significant. If this is mobilizing metals, mercury or other metals, this would be nice to know, and it might just be we need to be rethinking how safe sulfur is to apply. So I only have 2 studies doing this. I have we have this from Eve Link's study, and we have the experience in the Everglades, and we have all of the acid rain sites where this happened without intending. So those are my hydrologic examples. Now I'm going to shift to nutrients and organic matter, my favorite topics. So as you probably all know, there's huge amounts of non-point source and point sources of nutrients to waterways all across the country. This is an older plot showing large concentration from municipal sources, wastewater treatment plants, industrial processes, sewers, fertilizers, etc. And another plot showing how much nitrogen and phosphorus we're adding to the environment due to modern intensive agriculture. The question is, how do these high loads of nutrients affect the stream chemistry and the aquatic ecosystems? This is what I've focused my research on in most of the last 15 years, is trying to develop a portfolio of multi-isotope tools to be able to address questions of sources of nutrients, the effect of these nutrients on productivity in the water column, and how these end up sometimes causing environmental problems. From my perspective, it's changed from being interested in nitrate for itself to realizing that the nitrate is the cause. The nitrate, the ammonium, and the phosphorus is a cause of environmental problems that affect the people who live in the watersheds. So I think in a river to try to study the nitrate as though it's the environmental problem is a little bit blinders on, that most of the time the problem is actually ecological in response to the nutrients. That's where we're going for the rest of my talk. So the whole isotope mantra is, how do isotopes help trace sources and sinks of nutrients? And I've used this slide a zillion times, and it works because many times the different sources have distinctive compositions. Are they really, really distinctive? Are they moderately distinctive? It depends, but often enough it works that it's worth being your assume a spherical chicken idea. Give it a try. More often than not, it works. Isotopes are also useful for processes, and the interaction, the more the processes are recycling the nutrients, usually the more confusing it is to try to distinguish sources, but not always. So I'll have some examples. So, as I just said, this one is that the problem with environmental forensic studies with isotopes is the isotope fractionation. It can take things that were distinctive and make them not so distinctive. It can add a lot of environmental variability because all the little microbes in the soil and water are interacting in their own places. So, an important early study, I think I might not have mentioned that Tyler Copeland actually hired me at the USGS to set up a nitrate isotope capability at the USGS and use this new tool and capability to attack important environmental problems. This was in the aftermath of the Clean Water Act and one of the several amendments to the Clean Air Act, where these were important problems, and the USGS wanted to be part of developing tools and applications for this. This was a wonderful opportunity for me, but as coming in as a metamorphic petrologist, I barely knew what, the atomic weight of nitrogen, so it was a long pathway. At the time, I had a diary, and I wrote in my diary on that first day when Tyler took me in and showed me this broken mass spectrometer that was going to be all mine. I wrote in my diary, "Oh, my own mass spectrometer, just what every girl always wanted." And it was an interesting several years learning more about mass spectrometers, getting it working, and it took a long time to develop the method for O18 and Delta 15N that ultimately Steve Silva senior authored, and it's a method we used and for a short number of years dominated the field before people invented better, easier methods. But I want to start with this important study, Cole et al., because this study had a huge impact on isotope science in the United States. He was a published paper in Science, and it was rebutted by all the gods of the soil science community, basically saying you can't do this. This was the first study trying to use isotopes to answer the question of how much of the nitrate in agricultural streams might be derived from all of the fertilizer being applied to the fields. It was a sensitive topic. And this is a colorization of Cole's diagram, basically saying that the samples fell on a mixing line between a soil N-derived nitrate he determined by incubating soils to see what would form in as natural condition as possible, and the compositions of waters and drains and tiles. And I thought it was a pretty clean study, but the response by Huckadall made a number of points, kind of largely angling on not enough samples, too much environmentability, too much fractionation, yada, 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 all the things that can happen when you have a small-scale study, and there's always some place you could have sampled. I suspect if he had done what we end up doing 20 years later, sample at huge scales, large number of samples, these problems seem much less important. And that's where we are now. This study had a huge impact on nitrogen isotope geochemistry in the United States. It basically stifled anyone using nitrate isotopes for a surface water study for over 20-odd years until Steve Silva and I got our nitrate method working, and we used it in the Mississippi, and Bernard Meyer piggybacked on NASCON sites and analyzed samples and went from there. In the meantime, people were using the technique in groundwater that wasn't in the crosshairs of environmental interests. So, in the 20-odd years since we started working in the Mississippi, there have been hundreds and hundreds of studies using isotopes, and these kind of questions of, oh my gosh, how can you determine sources in the face of all this variability, just doesn't come up as often as before. So this is, thank you, Jeff, the Kendall plot, which is nice having Kendall plot rather than Kendall Tau, which is the usual thing I find when I go Googling for my name. It's statistics, but this is a diagram showing kind of the basic range of compositions of nitrate from different sources. It also shows an arrow going up to the right, which is what happens when you have nitrate uptaken by biota or denitrified, that it changes the isotopic compositions in a regular fashion. So the idea is you can collect samples, plot them on a diagram like this, and it's a starting place for interpretation. It's not a final place, it's a start, and go from there. So, low-hanging fruit. I think one of the real low-hanging fruit is there are a number of hydrologic geochemical models that are making predictions about sources of nitrate, phosphate, organic matter at large scales in the United States and elsewhere. And it just seems to me these are obvious candidates for testing, particularly when these models are being used for evaluating possible remediation strategies. It would be nice if the models were right. So testing models with completely different data is something which we should be doing a lot of. I'm not saying isotopes are the only best tool, but it's the tool in my toolbox, and it's something that can easily be applied. So I have several slides showing a model the USGS folks have been working on, the Sparrow model, and a lot of effort by the folks in the Office of Water Quality and the NASCON program has been on developing these models and then making predictions of the models at various scales. And so this is an example that the Sparrow model can calculate the total N and phosphorus, carbon, sediment yields from different watersheds. So, and then you can, that's not something immediately testable with an isotopes, but you can then use those same models to make predictions about where does that nitrogen come from. This is a model from 2000 from Smith and Alexander, and the idea is if you look at the 4 different maps, the things in red were where that source was 50 to 100%. So, for example, in the fertilizer plot in the upper Midwest, fertilizer is a dominant source of nitrogen. So those. Alexander et al. Have published a number of papers where they make these cute little pie diagrams of the relative percents of different materials coming in, and I look at these and go, well, if you collected isotopes at the same confluence area where these kind of samples, these kind of models were being calculated, you could test these predictions, because some of these different sources have distinctive isotopic compositions, or some of the sources are so small you can say, well, for a first cut model, we'll just ignore that one and worry about that on the second and third more complicated model. So an advantage of testing with isotopes, you can go out and collect a sample at any time or spatial scale you want to, whereas the models are calculated over very large time and spatial scales and cover a lot of environmental variability, whereas the isotopes, it's what was coming down the river then. So this is an example if you're interested in the percents of different N sources to the Gulf of Mexico. So I have used my delta 15N values published in a 2007 compendium, and what you're seeing in colors there is that the Animal and human waste, colored in orange and blue, have relatively high delta 15N values, making them different from crop agriculture and natural and rain sources of nitrate. So it'd be an easy test of these sort of models. You don't hear very much about phosphate isotopes. It's a relatively new tool. This is a set of data from my colleague at the US Geological Survey, Megan Young. She published a compendium of phosphate data from large part her own research, showing that phosphate derived from human waste generally has a much lower isotopic composition than other sources. You might be wondering, why are we not showing phosphorus isotopes? Well, sadly, phosphorus only has one stable isotope, so if you want to look at stable isotope ratios, you're using the oxygen isotopic composition. Rather analogous to we analyze nitrate for oxygen isotopes or carbonates for oxygen isotopes is part of the story. And it's also been used to predict the amount of organic matter to the Gulf of Mexico. And we actually have, Steve Silva and I, and Valerie Kelly published a paper on the Mississippi, and our rough numbers of organic matter sources match their prediction, which we were happy with. Anyway, more kind of models. You can use N15 and O18 that some, so this is using N15 of the nitrate and O18 of the phosphate, trying to get at the whole question of which nutrient is coming from what land use and what quantity, et cetera, et cetera. So that was a whirlwind through using isotopes to determine nitrate sources and testing models. I'd like to spend a few minutes on my probably favorite study of all time, which was the first thing I wanted to do when I first joined the USGS, when I was handed my chance to fix a dead mass spectrometer and use this for good science, is that I wondered, this was in the middle of the acid rain years when we were very concerned, where is the nitrate in rain coming from? Where is the sulfate in rain coming from? What's the contribution from coal-fired power plants versus vehicle emissions? And I thought, well, wouldn't it be cool if those different components had different nitrate isotopic compositions? And just the fact that I wished it'd be so didn't make it so, but it was enough of a hypothesis to go with, is I'll just assume that this might work. And it took about 20 years before we finally had all the pieces to make this work, which is a funding source, which was the New York State, NYSERDA, and the Electrical Power Research Institute. We had a new method that replaced our nitrate method that let us analyze much smaller nitrate samples, the microbial methods by Sigmund and Cassiati. So we didn't need very much water, and the realization that the NADP program saved archived water samples that could be tapped from this. And the fourth part is finding the right postdoc to actually do all the hard work of the project, and this was Emily Elliott, who was with us for 3 years and is now a faculty member at the University of Pittsburgh. This was a study that Beth Boyer, Doug Burns, and I put this together, and it's still my favorite study because the data are gorgeous. So I'm going to show you some. I have some plots from different things that Emily Elliott has co-authored recently, showing the whole cycle of the movement of NOx from the point sources, non-point sources into the atmosphere, how it moves around. Just a cartoon. So this was our design, and this was. I had to argue very hard with the funding agencies who kept saying, well, we're really only interested in New York and the Northeast. Why do you want to do this at the national scale? And why don't you want to go sample cars and sample power plants? Well, I was pretty darn sure that if you sampled the individual sources, you'd lose yourself in the noise of all the environmental variability. I wanted to do it at a big enough scale that the variability would be dampened out. It took a lot of persuasion, and they went for it. And so what we tried to pick is sites that had a reasonable distribution across the country. And so with the help of Mark Nillis and USGS colleagues, we picked 156 sites. We picked the year 2000. Mark Nillis helped us picking of the 5 years of archive samples what was the most average. And I was going to do one sample per one composite per sample, and I went to an NADP meeting, and they said, well, Carol, one sample there's a lot of environmental variability, temporal variability you might like to know something about. So I said, okay, we'll do 2 samples. Well, you know, summer and winter aren't the same in different parts of the country. And so before I knew it was 6 bi-monthly samples for each of the sites, and it was a huge set of samples to analyze. And the data are gorgeous. We've so far published several papers on various aspects of the study, but only 2 of them actually on the nitrate isotope data themselves, answering the original questions of the study. Could we use this to determine sources? And the senior author on all of them is a postdoc in the study, Emily Elliott, and they're really impressive papers. A 3rd paper is still in review and more to come. So this is a plot of just the January 2000 data. These are composited samples showing these gorgeous patterns across the country. This is the Delta 15N. And you click back to that one, you look at where the nitrate concentrations are high in the range sites. These are the sites where we're seeing the high delta 15N values that we figured would be the signature of a coal-fired power plant source. And the long and short of it is we were convinced with our set of data, empirical data, that you could determine rough values for the N members and use it to calculate percents to different sources. Subsequent to this, Emily Elliott, when she left the USGS and got her own isotope lab, she and a variety of excellent students have been publishing values on what are the actual M members if you sampled from tailpipes and sampled from this and that. And that's some of the data here from one of her students, Felix, showing the compositions of biogenic soil, livestock NOx, vehicle NOx, et cetera, et cetera. So a low-hanging fruit. We validated this at the empirical scale at this huge set of data, way more data than atmospheric modelers have and used to develop models suggesting you couldn't use isotopes for sources. And Emily and her students have done the subsequent studies showing that you sample the point sources close to home, they have different, that they are distinctly different. So this is at the point one could use this in places where people care and they're arguing about what is the main source of it and what should you remediate. How do you do this? It's a useful tool if you have 2 states nearby and they're futzing about whose NOx is causing problems. This is a tool that could be useful. So a useful forensic tool. This study fits the category of, I put in the category with the Cole et al. Study, where despite it being a convincing set of data on an important environmental problem, it's easy to think of all of the sources of variability which could make this not work. But it worked anyway, and we're working our way through convincing an atmospheric science community that were so cognizant of the importance of atmospheric processes that it seemed implausible that source signatures would remain. We've made the case with the N15, and we're working on making the case with the oxygen 18 that under some circumstances that the source signatures are not eliminated. Okay, so that was nitrate. As I said a while ago, most of our studies on tracing sources of nitrate in rivers is really at trying to answer questions about what is the cause of environmental problem. If the situation is hypoxia, killing off salmon, what is the organic matter that's the problem? If the problem is a food web collapse, how does this all play in the pieces of this? So these are kind of 3 general categories where we've used isotopes and they seem to be really useful tracers. One is the hypoxia, and this was why we got working in the Mississippi to begin with. And with Vance Kennedy's intervention, we tried to apply the same tools successful in the Mississippi, showing that a large percent of the organic matter, contrary to previous expectations, was actually algae and bacteria, not the inert, dead terrestrial matter that a lot of people think is the main source of organic matter to rivers. Same thing in the San Joaquin, it's mostly algae. Harmful algal blooms, I'm going to show you an example of that. It's one of my cool low-hanging fruit, is you can use isotopes, and we've done it and published it and have more papers in progress on using the isotopes to tell where is an algae or bacteria getting its nitrogen from. So if you have a microcystis bloom, if you can figure out that it's ammonium, not nitrate, then it helps you focus your attention on maybe wastewater treatment plants that need to be upgraded to nitrate or agricultural ammonium. And food web problems in the San Francisco Bay, it's been suggested that that collapse of the food web is a result of too much ammonium from the wastewater treatment plant, which causes smaller algal blooms that aren't sufficient to support the food web. So we're testing this with isotopes. So this I get into my multi-isotope, multi-component isoscapes along the river delta continuum and a chance to show a pretty picture. So this is an isoscape. You're more familiar with the isoscapes like the plot I showed earlier. That's a map of the United States with delta 18, delta 15N. This is a river isoscape, and I have huge sets of data collected along transects down rivers, and how do you plot this? And it took me a long time to come up with this, and I'm calling them eye candy because I found that they are incredibly attractive. People look at them and they could get this is a useful tool for understanding a source in a way they wouldn't get looking at my Kendall plot where they go, God, geeky stuff. So what this is plotting is the river mile between the part of the San Joaquin River that starts in the valley. San Joaquin River, that's the river part, and then the delta where the San Joaquin and Sacramento merge, and then through the bay to the Golden Gate Bridge at minus 45 river miles. What I'm plotting is the delta 15N in blues and yellows and delta 18O in pinks. On the y-axis is date. There are 1,000 samples analyzed here. I left off the points because I'll show them later, but this is pretty. It's like I like to make a stained glass out of this. It's so pretty. So what I made with these data is said, this is the kind of variability you're seeing. Our analytical precision is less than a per mil. You're seeing huge ranges of information that's sources and processes. So anyone who thinks, oh, it's just the same nitrate being diluted, you know, they're diluted. Sorry, bad pun. So I already said this one. So let me show you where the data came from. This is a map showing our sampling sites starting in the San Joaquin through the delta into the bay. These sites were sampled an extremely huge number of times. We started this study in 2005. We are continuing it now. It is, I am sure, the largest multi-isotope data set for a river delta estuary ever imagined, and papers are coming slowly. There's just so much data. We analyzed this for everything for nitrate, water. POM is particulate organic matter. It's what you get when you filter a sample under 0.7 micron glass fiber filter, and DOC isotopes. Smaller numbers of samples were analyzed for DIC, ammonium, sulfate isotopes, everything we could think of that seemed interesting. So let me start now that I've shown you the pretty thing and got your attention. This is what the concentrations of nitrate are about that got people interested in it. There's these huge changes of nitrate largely affected by flow. Where is the nitrate coming from? How does that impact the ecosystem? And those are the samples that we then analyzed for isotopes and could look at the changes in isotopes and interpret them in terms of sources and processes. This is that pretty isoscape again, now showing dots where the samples were collected on the upper panel. It's something 1,000 data points. We lack tools right now for the correct GIS plotting of data that are not isotopic. The waters are flowing. Processes that have their own non-isotropic nature, and we need people who could help develop how do you plot something that isn't introducing a implicit bias in it. So another room for GIS people to really contribute to this. I showed we've analyzed particulate organic matter for carbon, nitrogen. So any place you look at the values where you see a clump of values, for example, the blue values in the middle of a delta area, where suddenly you're seeing high delta 15N values, that is a hot spot. That hot spot happens to be where there's a wastewater treatment plant coming in at the city of Stockton. It caused a lot of nitrification, and it was the ammonium that was actually the source of the nitrogen to make the algae. You can tell that from the isotopes, whether the algae was growing on nitrate or ammonium. This was our first hint that this would be a useful tool, which I'm going to sip on and look at. So, a big picture. I think the most important part of our multi-isotope studies is this realization of how much of the particulate organic matter and now the dissolved organic matter is actually relatively new algae and bacteria. When I got into this whole study, knowing about the general consensus was these were big rivers, were too turbid to support much primary productivity, and so they were old terrestrial carbon entering the coastal areas. This has turned out to be wrong in every big river I have sampled from in the United States, and certainly smaller tributaries coming out of places with a steep hydrologic gradient, picking up a lot of sedimentary load. There's a lot of other things that can be in it, but by the large, despite what people thought about what the algae was doing, they were productive anyway. You might be saying, why do we care about the POM and DOM? This is why I got interested in this in the Mississippi is, yes, the excess nutrient is undoubtedly the main cause of algal blooms in the Gulf of Mexico and the subsequent decay of those algal blooms, consumption of oxygen and ecological problems. But there is fresh organic matter coming down the river that is contributing to it. It's a small part of the story, but there it is. And we determine the source of these by analyzing for isotopes, and I'm going to skip on through because I'm running a little low. Basic idea is there are different sources of organic matter. We analyze them for isotopes, and then with varying degrees of assumed spherical chicken type of models, calculate how much of it comes from different sources. We analyze everything for carbon, nitrogen, and sulfur now if there's enough material, because the sulfur isotope value is very useful in determining sources. There's actually a 35 per mil gradient of sulfur isotopes across this river delta continuum, which is a huge range of isotopes that lets you trace where things are happening. So, river continuum theory was something I learned about somewhere in an early biology course, and the idea, this is a diagram from it, I got this plot from Jacques Finlay a number of years ago, and kind of the idea was in very small rivers with overhanging canopy, there's not enough light, so of course there's very little primary productivity. You get to a certain size, it's big and open, you can get primary productivity, but when the rivers get to be really big, like the Mississippi, the idea was it was just so much particulate matter coming down the river, it wouldn't support productivity. But roughly half, if not more, of the organic matter in my 10 years of data from the Mississippi and all the big NASQUAN sites, it's algae plus bacteria. Why do I put the bacteria in it? Because with the techniques we're using, it's not an easy thing to try to separate out those two. But they're both aquatically produced, and from my perspective, they're close to being the same sort of thing. They're aquatic productivity. So this is a plot from the Mississippi. This is a plot that when I realized that one of the things I thought was true wasn't, that these rivers the organic matter was largely algae. So if this had been mostly terrestrial material, it should have been deadly dull and boring. What you're seeing there, particularly in the N15 values, is a huge range of compositions, meaning somebody was using nitrate or ammonium and using that to grow the algae. Well, if it was all coming from terrestrial sources, it would be mixing and looking like some mix of largely C3 plants and some C4 plants. Also, the C-Dan ratios are a dead clue that this is largely an algal material, bacteria. We have been hoping to analyze organic matter for dissolved organic matter also, and we have just recently, with the help of an Indian scientist, Atrulesh Kasiri, who wrote an Indian-U.S. Science and Technology Forum proposal to come work with us on a topic of mutual interest. He got a new method for analyzing DOM for carbon, nitrogen, and sulfur simultaneous isotopes, and we've been saving archive samples from all our sites, so I have 6,000 samples or something like that to use this on. Okay, in my last couple minutes, a really good low-hanging fruit, a topic of large interest, is these huge microcystis blooms happening more and more frequently in lots of places, including that very frightening one in Lake Erie in the fall, where people did not have a source of clean drinking water for a few days until the local municipality managed to clean the microcystis from their pumps. So this is a plot showing the green of the microcystis blooms in Lake Erie from August. We published a paper in 2007 with Peggy Lehman of the California Department of Water Resources, who's a microcystis biogeochemist, We were testing the idea that isotopes could be used to determine whether it was mostly ammonium or mostly nitrate that was fueling these blooms. So we piggybacked on the study, and we don't have ammonium isotope data for these samples because we didn't have a good method for analyzing ammonium at the time. And later on, we got a method. I'm not convinced the samples are the sufficient quality to analyze, and I'd much rather have no data than have bad data. So we just use the nitrate, but it turns out you don't need to have both of the isotopes under certain conditions. So let me show you this. So here's the general conceptual model is algae, and includes phytoplankton and cyanobacteria, can get their nitrogen from 3 sources. You can see a little diagram. And the idea is you could identify whether it's nitrate or ammonium if they're isotopically distinctive. So this is a cartoon showing how this works. So if the ammonium is in pink, the nitrate is in blue, their boxes are at different places. One of them has the ammonium has a higher isotopic composition than the nitrate. The idea is that if the algae are using ammonium, they use the light delta 15N of the ammonium preferentially, because this is what biota do. And the same thing for the nitrate. So you analyze the ammonium and the nitrate, you analyze the algae, and you see where it plots. If it's as a lower value than the presumed nitrogen source, it is consistent with that being a plausible source. That's different from it's just consistent and plausible. Fortunately, because I was pretty sure that isotopes are helpful, and if you try things out, that there's all this environmental variation, we started this before we were sure this would work. And this is a plot of proof of concept showing that, and again, it's river mile on the x-axis, various things on the y, showing downstream of a wastewater treatment plant, a pulse of ammonium coming from the treatment plant, and how that is slowly downstream nitrified to nitrate, and the effect of that on the nitrate and the ammonium N15 value. You can see that shortly downstream of the wastewater treatment plant, they become a few per mil different, analytically distinctive, and a few more miles down, they become whoppingly different. So this meant we could do this. So this is the kind of plot that I'm using, because I think it's kind of nice and graphic, is you plot the algae or the organic matter, whatever it is, the delta 15N on the x-axis, you plot the nitrate or ammonium on the y-axis, and you see where they plot. The biota has to have a lighter isotopic composition or equal to its nutrient source. That's the way isotopes work. The light materials are concentrated in the product. The heavy materials are retained in the reactants. So you plot your data. If they plot below the line, its significant uptake of local nitrate is very unlikely. It's implausible. If they plot above the line, it's plausible. So we show the data. So this compares the microcystis samples. They plot below. And other sites, Sacramento River, the San Joaquin, that we knew it was dominated by nitrate. It's kind of proof of concept that the microcystis sites predominantly plot in an area saying nitrate uptake is implausible. Could it still be possible that nitrate is a dominant source? You have to imagine that the microcystis was collected in a place not where it grew, and microcystis can be blown across a waterway, and so it could be separated. It's something still to be worked on, but this one says at a first cut, this is implausible. This was part of a huge study with isotopes and modeling, whatever, so we had lots of different parameters that showed that not only could we say that it was not nitrate from the San Joaquin, it was not nitrate from the Sacramento, and you're left with, well, if it's not nitrate from either of these, where the ammonium could be ammonium, and the statistics work with the ammonium coming from the wastewater treatment plant being a source. So a couple more minutes. In most places, it seems to me that people are thinking that, maybe that's overstated, in a number of places, people are thinking that phosphate, not ammonium or nitrate, is the nutrient fueling the bloom. Can you use phosphate beta isotopes to answer questions in the same sort of way? This is a set of data from Megan Young in my group. She analyzed these as part of a PhD project, showing that organic fertilizer and wastewater treatment plants and detergents have moderately distinctive compositions. You might go, oh, these aren't very different. This is as good as N15 of nitrate was at this stage in its use as a tool. This is very promising. A problem with phosphate O18 that doesn't affect nitrate O18 to anywhere as much is the phosphate-oxygen exchanges. As phosphate is cycled through the ADP cycle, every time it goes through, an oxygen has a chance to exchange with the water. So this technique, you're more likely to be able to determine sources if you have high concentrations of phosphate in a situation where not being recycled too quickly. This was one of our field examples. This was part of Kara McLaughlin, one of Adina Paitan's students. Megan was also one of Adina Paitan's students, showing in the San Francisco Bay, we collected phosphate samples and showed that most of the samples could be explained by a simple mixing of a delta N member and a seawater N member of phosphate, but there were several of them that plotted below the line saying there was another source of phosphate. This is a tool that's a usable tool. So a low-hanging fruit here. There are lots of places that are experiencing microcystis blooms where reliable information about the type and geographic source of the nutrients that fuel the blooms could be very useful for developing sensible remediation strategies. So this is a technique people can go develop a hypothesis, make a sampling strategy, and go for it. And I've run out of time, so I'm just going to a couple little pontifications, things I learned from our 25 years of piggybacking isotope studies onto all sorts of programs. Biggest problems? Yes, the sources have partially overlapping compositions, a lot of spatial and temporal variability, isotope fractionations have a problem, blah, blah, blah. Our approach is multi-isotope, multi-tracer approach. Collect lots of samples, don't analyze everything at once, but if you get to the point where you don't have enough you can't write enough equations for all the variables you have something else you can analyze. An interesting observation that I've touched on is a number of these studies have shown that this idea of sampling at what is the proper temporal scale for addressing the question you're interested in. And I and my colleagues have been very fortunate that these big scale studies have convinced us and the reviewers of our papers that you can empirically answer questions about sources in a different way than you might normally think of in doing samples where you actually collect all the N members. So good examples of how this is a powerful approach are at the bottom, that we find that atmospheric processes are not entirely overprinting the NOx isotope source signatures by these big scale studies. That's our study, the various papers by Emily Elliott et al. Soil processes are not overwhelmingly overprinting nitrate source compositions. This was the Cole et al. Study. The criticism by the soil science community stopped people from doing studies like this in the United States and streams for about 20 years, but over the span of time, Cole has been proved right. He had a tool, he was using it correctly, and this was an approach that should have been used in surface water studies much earlier. A third one is the food webs. A lot of people, biologists early on, 20 years ago, found it offensive that the isotoper wouldn't be the least bit interested where the bug was coming from. We just wanted to dry it, grind it up, analyze it for isotopes, and tell what the bug was eating rather than care the feeding studies. Well, study after study have shown that you don't have to know that kind of information to make first cut sensible models. So the right tools at the right scale. Why piggyback studies? Let's us do bigger studies than we could have done if it was just our own resources. USGS has been very nice letting us piggyback our studies on large monitoring programs, and they have been cooperative in letting other people piggyback on. So if you think of something interesting, it's worth asking. People are more likely to say yes if you make it easy for them. Tyler Copeland and I sent off bottles and coolers and pre-made out sampling labels, and this persuaded the NASQUAN people to save us samples. For years, people collected me particulate organic matter samples because Rick Hooper, when he was in charge of the NASQUAN program, worked this out, that they were going to filter through their waters anyway. Why not just not throw the filters away, but fold them, freeze them, send them to Carroll? It was no extra effort on their part and allowed all of these subsequent studies on the importance of organic matter to come to fruition. It was a simple conversation in a field vehicle with Rick Hooper that ended up leading to all of this work, and the people who were willing to collect me samples. More isotopes, the better. Multi-isotope studies, I think, more chances to answer the question. And again, for the sixth time, I think the biggest bang for the buck in an isotope tracer in a river where you're interested in ecological problems is it's collecting and analyzing POM isotopes. It's way easier, cheaper, faster than nitrate isotopes, and you get a large part of the story for something really simple. You can do vast surveys of just filtering your waters and saving the filters. And again, archive, 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 and archive. I learned this early on from Vance Kennedy, who our first study in the mantle only worked because he had archived samples from the previous storm that when we were wondering about was soil water a contributor to stream water, he had the rainstorm that filled up the soil zone with isotopes. Brian Bergamaschi taught me this in the San Joaquin because when I suddenly got interested in nitrate isotopes, he had frozen water samples. You can test a lot of hypotheses with this. So I've been putting versions, this is my last slide I think, versions of this slide up my whole career. Why we use isotopes. It's one of many tools that can be used for water resources management. I'm not saying it, I talk about isotopes because that's what I'm doing. Lots of other tools, more tools the better. And I used to say the value of it was the first one, we often learn things, blah, 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 blah. And I would say at the bottom, more likely that any proposed remediation plan will be effective. What my studies in the last 10, 15 years have shown is really the important one is number 2, is you learn things about water resources that contradict what you think you knew before. That's really so much more important and so much more fun coming in with isotopes to the rescue, poking holes in everybody's theory. That's really the most fun part. It's surprising how often this is true, and it supports this contention that I like this little phrase as half of everything you think you know and everybody else thinks they know might not be true and should be open to the possibility that it's a big world out there with lots of fun voyages to discovery for us all. So, thank you. Questions?
Carol, that was fantastic. There's a prize for those who can guess how many PhD theses were given there. 30, 35, it's clear you should have been in a university. You wasted your career at USGS. There's many, many, many students who would have benefited by having PhDs mentored by Carol. Questions? We're going to take a few minutes for questions if there are some. Yes. Speak up loudly.
Based on your San Joaquin work, yes, based on my San Joaquin work, I wouldn't extrapolate it that far. We only have one microcystis study where we've done isotopes, and I.
So, what would you say they should be doing to properly attribute their outbreak to potential sources there?
See if anyone had archived samples, for one, time machines being too expensive to afford. Start sampling now. Get a routine monitoring program. Go and archive water and the filter you filtered it with. These don't take up much effort, and 2 years later, when something happens, you have the samples. Cheap, simple tool to answer things that are really important and controversial. Phosphate is more trouble. It takes big samples, more futs, more bother, but there is not a study showing that approach works on microcystis, but this is a low-hanging fruit, a place where people think that's the source. It's a testable hypothesis. Collect the samples to address the other possible sources. Low-hanging fruit.
Your sulfur method for fingerprinting flow paths be used in the forest to look at preferential flow through?
I think I've seen things like that from Bernard Meyer and Myron Mitchell with their sulfur studies and whatever. Not necessarily in the bypass flow, but it might have worked there and we just didn't know to look at it. So quite possibly. It's because the real key was the different flow path that the sulfate goes through. If it goes through the soil, it goes slowly, more reaction. If it goes through the bypass, it doesn't get evolved. So I think this is a possibly useful lots of places.
Carol, we have to remember that this was a lecture to put in perspective some of the work that Walter Langbein did. And I think Walter, if he was still alive today, would applaud the integrated work that you've done over a career, taking a technology that had some stopping points, leading in there, showing us new ways to do things. So thank you very, very much.
Thank you. That's very nice of you.
If there's no other questions, then, oh, there's one down here, then.
I might try one out of ignorance on your delta in the lens, I think. You showed the whole country plot, and it looked like Los Angeles was the source. When he made it, pull that slide back.
We can come look at it. But some of those coastal sites, there's just one sample, and you wonder if there'd been a second sample if it would have shown the same value. Secondly, there's not a lot of rain in Southern California most of the year, and so it was a very small. Not every sample was, not much rain that came in, and often there wasn't enough rain to analyze. It's an extremely biased data point. But further work. Let me just throw in here, since I still have some of you folks here, is these ideas of low-hanging fruit. A lot of them are things that I'm really passionate about, and if I was a faculty person and had lots of NSF money for students, so I would try to interest them in a bunch of these. But there are a bunch of you out there who may have your own source of funding. You might dream up a funding source to write a joint proposal on where we are open to suggestions of collaboration. This is a fun field, and the more fun things we do, the happier we all are.
So I spent a year at the USGS back in Reston in the mid-1980s reviewing the NASQUAN program. And at that point, the powers that be really wanted to get rid of it because NAQUA was just on the horizon. And the view was it was kind of mindlessly collecting, monitoring data that no one was really analyzing, and they really needed to do more process work, and NAQUA was going to do process stuff. So I was very interested in the studies that you piggybacked, as you said, on the NASQUAN data. But now looking back at this 30 years later, do we have an adequate basis to do sort of national scale studies with NASQUAN having basically, I think, more or less vanished, and the NAQUA strategy, which was supposed to rotate every 6 years or something and come back and look at things and so on. We have several decades of that. Is that working out?
There just isn't much federal funding for monitoring programs. It's unsexy, and when we have a disaster and it's USGS monitoring data that explains flood problems or something like that, the more money trickles in. But I was hoping that the Secure Water Act, which mandates some huge number of gauge sites, that this might move itself into more places where people are collecting samples. Or maybe this needs to be crowdsourced, I don't know. But my answer, I wish they had kept the NASQUAN program going, because there were a lot of people who paid attention to it, and there are big rivers that are interacting with the ocean, and we really, that they're not being investigated. So.